Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 24th December 2020. As all of us know, we are into the revision mode and we are revising the entire syllabus. As part of it, we are into the 18th chapter, that is body fluids and circulation. In our previous session, we talked about the connective tissue called blood and what are the parts of the blood, what is the significance of RBC, WBC, platelets as well as plasma. And then in this session, we are going to talk about blood groups. Okay, so how blood groups are formed, how many blood groups are present in human beings, and what is the basis for that blood group. So these are the things that we reflect upon in this session. As all of you know, we must be thankful to one gentleman called Landsteiner. He is credited with the discovery of blood groups in human beings, and he has reported A, B and O blood groups first time in human beings, which are determined by a gene. And as all of you know, the gene is called I gene. Okay, it is, we just write I, capital I gene. What do you mean by this I gene? Isoagglutinin. I for isoagglutinin. So this gene has got the three alleles. You know, already we talked in genetics chapter what is a gene and what is an allele. So the gene has got the three allelic forms, and those allelic forms are IA, IB and I0 or IO, we say. Okay, these are the three allelic forms, and these alleles proteins they produce the proteins, and the IA produces, allelic form IA produces a protein called antigen A, and B produces antigen B. These are proteins, okay, and IO doesn't. It doesn't have any protein, any antigen. That's why we are writing here, look at that table, nil. Antigen is nil. So when we put this in group form, we have got basically four groups, and the four groups are blood group A, blood group B, blood group AB and blood group 0 or we say O. Okay, and what would be the genotype of this? For blood group A, it has to be IA. Okay, this is one form, and if another form is I0, this is the genotype. And remember from genetics chapter, when we say genotype is the genetic combination. Genotype is the genetic makeup of an individual or gene combination. So the gene combination, allelic combination is, if blood group has to be A, the allelic forms that are to be present in the blood is IA or I0. Then the blood group becomes A. And the antigens, where are they present? What is an antigen? Antigen is basically a protein. Where is it present? On the surface of the RBC, red blood corpuscles. So if antigen, it's a protein, if it is present on the surface of the RBC, then it is A antigen. Just why I said IA, it produces a protein, antigen A. That's why we are saying antigen that is produced is A. And what about antibody? Antibody is also a protein, but only difference is it is present in the plasma. And what is the antibody that is present? It is B. I can say anti-B, or we can use, just for clarity, I will write small letter B. So there will be always a fight between antigen and antibody. Okay, and this person belonging to blood group A, he can give blood to A and AB. He can give blood to A as well as AB. Why? Compatibility is there. And he can get blood from A or O. So he can get blood from A or O. Why O? Because antigen is nil. No antigen is there. So the antibody antigen fight will not be there. That's why we say O is universal donor. So the donors group is A and O. What about next group? Blood group B. How this is formed? The genotype has to be IB or I0. This is the genetic combination. If these allelic forms are there in a human being, then it is blood group will be automatically B group. And in the RBCs, on the surface of the RBCs, there is a protein antigen B. And in the plasma of that person, another protein called antibody that will be anti to this. So I will write it as A. So there will be always fighting between the A versus B. And to whom they can give blood to? They can donate blood to, because B blood group, they can donate blood to B and AB without any problem. And from whom they can get blood from B, blood group B and O, so that there is no problem, compatibility will be there. What about blood group AB? The genotype has to be, because both A and B are present, the allelic forms should be IA, IB. This should be the genotype of a person. These are the genes that should be present in that human being. Then that human being will be belonging to AB blood group. And antigens, of course, both antigens will be there. Antigen A as well as antigen B. That means two proteins are present on the surface of the RBC. And antibodies, no antibodies in the plasma. So that is the reason why they are accepting. They accept from all. That's why we say AB blood group is, look at that, he can take blood from A, he can take from B, he can take from AB of course, he can take from O also, just because the antibodies in the plasma are nil. So AB we say universal acceptor. He can accept blood from anybody. So AB group is called universal acceptor. From all he can take. But to whom he can give blood? He can give only to AB, because of, again, compatibility. What about O? The allelic forms, the genetic genotype should be IO or I0. That's why the blood group is O. And antigens, as we said repeatedly, no antigens on the RBC. RBC surface should be smooth. And what about antibodies present in the plasma? Both the small a as well as small b, both will be there, antibodies. And can give blood to all. They can give to everybody. That's why we say blood group O is universal donor. He is dana karna. He will donate his blood to anybody, universal donor. But can get blood, from whom he can get? Very unfortunate. He has to get from O only. He can't get from others. Why O only? Because of the antigens being nil. If he takes blood from others, there are antigens A, B, so there will be fighting between antigen and the antibodies. As a result of fighting between the antigen and the antibody, what happens? Agglutination takes place. That's why the word agglutinization means blood clotting will take place. So to avoid that blood clotting, compatibility should be there. So whenever there is accident or injury takes place and person requires blood, it compulsorily one must check. Every person, particularly biology students, we all should know what is our blood group, so that in case of an emergency, we know from whom to take. Like we said, blood group A, he must get blood from A and O only. Blood group B can get blood from B and O only. But AB people are lucky, very lucky, because they can get blood from anybody. So only universal acceptor. And blood group O, of course, they can donate. As I said, they are universal donors, they are karnas. But then when they have to get it, they have to get blood from O group only. That is the issue. So you need to check whether you are getting blood from O or not. If you get blood belonging to non-compatible group, then there is a severe problem. Agglutination takes place. Blood clotting will take place. Internal clotting takes place. It may lead to death. Hence, one must know his or her blood group. If you do not know as on today, I advise you all to get the blood test done and know your group. So during blood transfusion, blood donor has to be matched. Look at that. Blood donor has to be matched with blood of recipients to avoid clumping of RBCs. RBCs will be clumped. Group O blood can be donated to any individual with any blood group, so it is called universal donor. Person with AB blood group can receive blood from any person of any group. Again, why any person of any group? In AB, antibodies are nil. So, AB people are called universal recipients. They can get blood from anybody. They receive blood from anybody. And in that, there is one more protein that is present in the blood, that is called RH. It is Rhesus Antigen. Like we said, antigen A, antigen B, two proteins are there in blood. Another one, second one is the Rhesus Antigen. So a protein named as Rhesus Antigen, RH antigen, is present on the surface of RBCs in many persons. When the RH negative mother is pregnant with RH positive fetus, RH antigens of the fetus do not get exposed to RH negative blood of the mother. In the first pregnancy, as the blood of the mother and blood of the fetus are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of maternal blood to small amounts of RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, first time no problem, subsequent pregnancies, the RH antibodies from the mother, that is RH negative, can leak into the blood of the fetus, which is RH positive, and destroy the fetal RBCs. This could be very, very fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This is what we call it, erythroblastosis. So RH is basically Rhesus antigen. So RH antigen similar to Rhesus monkey. You know, there's a monkey called Rhesus monkey. So from that, it is expected that this protein has come to us. So this is observed on the surface of RBC of majority of the individuals, about 80%. Such people are called RH positive. So if RH antigen is present on the surface of the RBC, that person is called RH positive. Suppose A blood group. And if RH also is present in that, then we write A plus. If it is not there, then RH negative, A minus. So person having A blood group will be of two types, A positive and A negative. A positive means basically is belonging to blood group A. In addition to the antigen A, there is antigen RH. So two proteins are there on the surface of the RBC. Then we say A plus. If only antigen A is present, but RH antigen is not there, then we say A negative. Similarly, B positive, B negative, AB positive, AB negative, O positive and O negative. So here O negative has to be careful. So this is how we make 4 blood groups can be into 8 types. So that is A minus group, A plus group, A B plus group, B minus group, AB minus group, AB plus group, O plus and O minus. Do you know to which category you belong to? If you know, fine, good. Note down in your personal diary or in somewhere in your personal files. And then what we said just now, they based on the presence of Rh factor like erythroblastosis fetalis can take place, which is very dangerous to both mother and the child. If father blood is Rh positive and mother blood is Rh negative, the fetus blood will be Rh positive because law of dominance. Father's blood is Rh positive means Rhesus antigen is present. During the delivery of first child, there is a possibility of exposure of mother blood with fetus blood. Mother blood Rh negative, fetus blood Rh positive. So this develops antibodies in the mother blood. In the subsequent pregnancy, the mother's blood can leak into fetus blood and destroy the fetus of RBC. So this case is called erythroblastosis fetalis. And what about blood clotting? Blood clotting at the site of an injury, like whenever you are playing games or doing some work, accidentally, the injury may take place. And what is the common observation? When injury takes place, blood starts oozing out. But also you must observe that after say 1 minute or say 30 seconds, the blood stops oozing out. Blood starts clotting. Clotting of blood takes place. Thanks to that, it should happen. But in some people, it will not happen. The blood will be flowing continuously as though you have opened the tap, water goes out. Like that, blood goes out continuously. That's very, very dangerous. Thanks that blood is clotting so that we are not losing much blood. How does that clotting takes place? That's called coagulation of blood or blood clotting. When an injury is caused to blood vessel, bleeding starts, which is stopped by a process called blood clotting. An injury or trauma stimulates the platelets in the blood to release certain factors that activate the mechanism of coagulation. Calcium plays very important role in blood clotting. So at the site of an injury, the blood plates disintegrate and release a phospholipid called platelet factor 3. It is called platelet factor 3. Let me write platelet factor 3. This is the first one that is released at the place of injury. Platelet factor 3. And this is normally we say platelet thromboplastin. Injured tissues also release a lipoprotein factor called thromboplastin. Look at that, thromboplastin is released. These two factors, that is platelet thromboplastin and lipoprotein or thromboplastin, combine with the calcium ions. Look at that, Ca2 plus. So these two and certain proteins of the blood plasma to form an enzyme called prothrombinase. This is most important. Prothrombinase is the enzyme that is present. What does it do? Prothrombinase enzyme breaks down prothrombin, that is inactive plasma protein, into active protein called thrombin. Prothrombin becomes thrombin. Okay? And when the thrombin is very active protein and in some small peptide fragments also will be formed. Thrombin brings about depolymerization of fibrinogen into monomers. Later, thrombin stimulates repolymerization of these monomers into long insoluble fiber-like polymers called fibrin. So fibrin will be formed. And the thin, long and solid fibers of fibrin form a clot that seals the wound and stops bleeding. That's how clot takes place. So this is how an enzyme called prothrombinase, thanks to that, it plays a very important role. Hence, this is the mechanism of clotting, blood clotting. So we'll talk in our next session another liquid, that is lymph. How exactly the lymph